Antibody-mediated immunity is carried out by B lymphocytes, or B cells. These cells differentiate into antibody-secreting plasma cells in the lymph nodes and spleen. The body contains millions of different B cells. Each has on its surface a unique form of the receptor. The receptors are immunoglobulins, very similar to the antibodies that the daughter plasma cells will secrete upon activation. Receptors are produced by genetic recombination. Millions of different variations of receptors may be created. Each of these B cell receptors is capable of binding to a specific antigen. Unlike T cells, B cells bind to unprocessed antigens. However, B cells are most likely to become activated when co-stimulated by nearby T cells in lymphatic tissue. B cells can act as antigen-presenting cells. They incorporate the antigen and digest the antigen. Vesicles containing peptide fragments fuse to vesicles containing major histocompatibility complex II molecule. The fragments and molecules then bind together. The vesicles undergo exocytosis and insert antigen MHC II complexes into the membrane. Then, a helper T cell with the appropriate T cell receptor binds to the presented antigen. The binding of the helper T cell with the B cell triggers the release of interleukins, which causes activation of the B cell. The activated B cell proliferates into many identical B cells in a process called clonal selection. Most of these cells differentiate into plasma cells. These plasma cells are effector B cells, which release antibodies. The antibodies are able to bind to the same antigens as the receptor on the original B cell. Some of the B cell offspring do not become plasma cells. Instead, they become memory B cells. These cells can remember this particular antigen and will quickly respond to it in the future. The end result of activating the B cells is the production of antibodies. These antibodies help fight infection in several ways. Antibodies can neutralize antigens such as viruses and bacterial toxins by simply coating them and blocking their activity. Antibodies can attach to bacteria, preventing their movement and limiting their ability to spread to surrounding tissues. Antibodies can form large antibody-antigen complexes which will agglutinate or precipitate the antigen. These complexes can then be removed by phagocytes. Antibodies initiate the classical pathway of the complement system, which produces inflammation and can lead to the death of the microbe. Antibodies can surround microbes. In a process called opsonization, their constant chain regions enhance phagocytosis.